All right, guys, we're gonna work on chapter 3. See how far we can get with this. I'll try to break it into little chunks so you don't have to sit for a couple hours at a time. We'll take a couple little breaks. But this is chapter 3, applications of antigen-antibody reactions. And what's ahead? Here's our what's ahead page. Every blood bank test performed is looking for this antigen and antibody reaction. And as we discussed previously, this reaction is going to appear to be agglutination. So that's what we want to see when we see an antibody. Antigen reaction is agglutination. Let's get some light on the subject here. There we go. One thing we're going to talk about particularly is that anti-human globulin. Now remember, this is actually an IgG antibody. Okay, so this is a manufactured antibody that is targeted against any human antibody. So yes, it's an antibody to an antibody. And that's because this antibody here is just, we have to consider it just a foreign protein. So it's just a protein, okay? So this, we'll work on this, don't worry. We'll talk about it quite a few times, okay? All right. Again, here's some of our history. We can see that ABO, this was really important. Been around 120 some years now. Pretty cool. Here's some of our additional blood group antigens. The other day I mentioned in class that there are over 300. Okay, and we're just going to talk about a few of those throughout this course. Okay, anti-human globulin. This is a very important technique. Okay, and then the CAL system. One of the things I keep hearing over and over again with some of the podcasts and so forth, and the extra education I do with blood bank, one of the mnemonics that's used, so the CAL system. So CAL is another one of those antigens that are on the outside of a red blood cell. So I'll just draw a little K on there. There's more than just big K or CAL in this system, but CAL is the most significant, big K. And anyway, the mnemonics that goes along with this is if someone has anti-CAL, CAL kills. Okay, so it is very imperative that we identify the antibody CAL if someone has it, because it can lead to patient demise. Okay. Here's a little bit of our review. Immunoglobulin functions. Okay, if we think about this, here's our antibody, okay, or immunoglobulin. It combines, here's our variable region right here, with an antigen, okay, and that's where it binds. So antibodies bind to antigens, okay, and those lead to a variety of, this one says effects, but it leads to, phagocytosis by different cells, macrophages, okay. It can lead to the complement activation, which will lead to lysis, okay. And we can have extravascular hemolysis, okay, which gets removed by the reticuloendothelial system, okay, which is mostly your spleen. Okay, there are other organs in that, but your spleen is the biggest one there. Okay. Adaptive, the adaptive part of the immune system and these antibodies are when we have memory. Okay, this is where the chicken pox comes in, COVID, our flu vaccine. That's how it works. So it has adapted. We've made antibodies specifically against it. And a lot of times I'll make my chickenpox antibody and it looks like I put a little beak on there, okay, when I have drawings. Now remember, immunoglobulins are proteins, right? How many chains? We have our light chain and our heavy chain. How many types? We have 5 isotypes. Which ones will we focus on in blood bank? We focus on IgG and IgM, okay? Which one shows up first? IgM will show up first. And of course, you only have one other option for which one shows up second, our IgG. Now, after re-exposure, which one rises first? And our IgG is that one. More review, IgM. This is the first one, produced in the primary response. We call this a direct agglutinant. Okay, so this is the one when we mix our reagents together in our tube, the IgM is the one that we're going to see those clumps. We're going to see those aggregates. Okay, so when you were in lab and you saw those clumping cells, it was the IgM antibodies that does that. Okay, it can bind multiple red blood cells, and that's what makes it so good at this, because remember, it's got 10 binding sites, so it can make that lattice. Okay, that's our valency, and it is 10. Good things to remember. All right, and here is a nice chart, and this does talk about our heavy chain and light chain. Okay, and that's, remember, our heavy chain was through the middle, and the light chains were there, okay, from the other image we had the other day. Excuse me. Okay, so what else do we want to remember from here? We know IgM is huge. Okay, comparing those, valency, 10 versus 2. Total concentration, IgG, we have the most of it. Okay. Half-life is kind of fun. Okay, 5 to 6 days versus 23. Again, that's not something you have to memorize for the test. Right here, this is huge. Okay, make sure you know which one crosses the placenta. And I always think about the M just being larger. It can't cross the placenta, okay, where our IgG will. Okay. Complement activation. They both can activate complement. IgM is much more efficient at it and will cause that intravascular hemolysis. And remember that intravascular hemolysis, if these are our veins and arteries right here, veins and arteries, which is our vascular system. Okay, that hemolysis is happening. Our little red blood cells are rupturing in the vascular system. When we talk about extravascular, we have our little red blood cells, and they have antibodies attached to them, but they're being removed by the spleen through the reticuloendothelial system. Okay. Optimal temperatures. This is also important right here. This is room temperature, and this is 37. All right. Agglutination and immediate spin. Yes. No. IgM, agglutination at the antiglobulin test, and that's what we'll learn more about. No, but we will with IgG. Okay. Okay, IgG. IgG is our most abundant class of immunoglobulins. This one's showing 75%. I think back here, if we look at our chart, it went 75 to 80. So just be aware, you might see 80% on a quiz or test, and be prepared for that. Okay, but it is the most predominant one, most abundant. it is produced secondary immune response. Valency, we've talked about, is 2. Okay. There are different allotypes, of course, within the class, and we're not going to really focus or get into those, but beware, there are additional ones. So some of our testing techniques, and you will hear these different words, and we need to become familiar, accustomed with them, and understand exactly what they mean. So we have an indirect antiglobulin test, IAT, and you might hear someone use an IAT. And then we have the direct antiglobulin test. And this is, I typically call it a DAT. From what I understand, there are some individuals that have just called it a DAT. Anywhere I've worked, that's not been a common term that's used. But the difference between these two tests specifically is the in vitro versus in vivo. Okay. Now, when we talk about a DAT, and we're going to go over this multiple times, okay, this detects red blood cells, okay, that have been sensitized in vivo. And when we talk about in vivo, vivo is inside the human body, okay. In both of these tests, we're going to use AHG, okay, and we're going to go way more detail in this and work on understanding it, okay. The indirect, and just think about these words, indirect, okay. Indirect is always outside, okay. It's different, and so indirect is when we have the in vitro, okay, reactions or sensitization that we're detecting. And in vitro means in the test tube. And so we are instigating this, all right? When we talk about this one as in vitro, okay, or in vivo, I'm sorry, in vivo, I'm underlining that. In vivo, this is in the body, okay? In the body, okay? And again, I have lots of pictures we're going to go over with that, okay? And one way that we can see this is when we are looking, here's our in the test tube, okay? And we add our reagent red blood cells. There's our red blood cells. And then what we're doing is we're adding patient plasma. And in this plasma, let's make a bigger picture here. There's our bigger drop, okay? We have lots of antibodies in this plasma, okay? And oh, there's our beak for our chickenpox one, okay? That's actually not what we're looking for in blood bank though, but they're in there because it's plasma. There's all sorts of things in the plasma. Okay, there's glucose in there, C6H12O6. There's, electrolytes in there. This is straight patient plasma, so all these things are in here. But what we're looking for is we want to see if any of these antibodies in here are going to attach and react with these red blood cells. Okay, so we're adding this plasma and it's mixing, okay, and we incubate it and we do all those wonderful things, add enhancement media to make it react better. And so we want to see if any of those antibodies are specific to these red blood cells, okay? And these are going to be IgG, typically, what we're looking for at this point in time. But now we also know when this happens, that IgG is so small, okay, it doesn't cause that direct linking or lattice formation. So we can't see it because the IgG is so small. These are IgG. Okay? Remember, our IgM are really good at it, and they lead to that direct agglutination. Okay. So in order for us to see this, this is where this anti-human globulin comes in back there that we briefly talked about. And I'm going to make it this green. Okay. We're going to add that anti-human globulin, okay, and it looks like this. It's green. Oh. And what it does when we add it to this tube, so this is our tube again, right? And so we're adding a drop of this anti-human globulin. It will bind here and here, and it creates the agglutination for us to see, because this anti-human globulin is an antibody to human antibodies. Okay, we'll go over this more. Okay. Now in the DAT, what we're doing here is, here's our test tube, and remember this is direct. Okay, over here this was indirect, because we added everything in the test tube. We had reagent cells down here and patient plasma here. So this was patient, this was reagent. Now with a DAT, over here we're adding patient cells. And we're looking just at these patient cells to see if they already have an antibody on it. Because remember, we're determining to see if the antibody has attached to those red blood cells in vivo. So we're not adding an additional antibody step here. We're just washing the cells. But what we do next is we add our AHG reagent. Okay. And so if there are antibodies on these red blood cells, that AHG reagent is going to bridge those for us. And so we see that agglutination. Okay, that's our DAT. Again, we're going to go over this more. Okay. So, anti-human globulin reagents. Remember, this is the green antibody here. And this is a special antibody that is against human immunoglobulins. Or human antibodies. Okay, how do we get it? That's what's fascinating about this. So originally it was made by injecting an animal with the specific human protein, which is the human antibody. We're going to make it yellow. This is our human antibody. Okay. And then that animal, because it was a foreign protein in their system, the animal made an antibody to it. Okay. And then what they did is they harvested that and utilized it. Okay. Now, a lot of times what we're doing is we're taking cell lines that produce an antibody. Okay. And so we're programming that cell line to make the specific antibody. And the cell lines would be B cells. Okay. And this is where we've utilized those cancer cells, okay, because they continue to reproduce. They don't die. Okay. They live infinitely, so to speak. And they're identical because they're B cells. They just make the same antibody, exactly the same. And we utilize that then. And this is a much more pure, okay, reagent, much more cleaner way to do it because it's just the cell lines. We don't have all the extra proteins from the animal and so forth. There can be a lot of different variations in how the animal makes the antibodies, just like humans. So the cell lines make us much better reagents. And here's the advantages of some of these. And we do call these monoclonal. And it's unlimited production. Okay? There's very small variation between those. Okay. No human or animal sources, and no contaminating antibodies. So up here with this one, we have to purify it and remove it from all those other antibodies that the animal might have. Disadvantages. It may just be a little bit too specific, okay? And I haven't found that in practice to be too much of an issue, okay? And that's where it is that single specificity. But they've done a good job making these that we don't really have too many of those issues, okay? So we have monospecific anti-human globulin, okay? And we have polyspecific. And what that means is, let's just think about mono. Mono is 1. So we have a reagent that is either IgG or our C3b. And C3b is one of those complement molecules. And we'll talk about why it's important to test for both of these. And then polyspecific is many. So this polyspecific AHG has both. Okay, so it's both. Okay. The AHG must at least contain anti-IgG for antibody detection, okay? And for our pre-transfusion testing. Okay. What you're going to find in your reagent racks is polyspecific, which is what we're using. Okay. Our indirect antiglobulin test, okay, which you're going to do next week. We'll do it in lab. It is a 2-step method, and this is where we have our tube. We add our reagent cells, and then we add the patient plasma. So these reagent cells, remember, we have to have something known, and so these are reagent cells. Those are reagent cells, and we add, that's not a very good drop, did I make that? Let's at least make it look a little more like a drop. Okay, that's our patient plasma. And this, we're going to incubate it, add enhancement media, and give that the opportunity to attach if there are any antibodies present. We really don't want antibodies present, so we want a negative reaction. But in order for you to learn what's happening, I'm going to show you that there are antibodies in there. But we would prefer negative, no antibodies present, okay? So we're giving that time to attach. And when I say 2-step, that's where now we're going to go. Here's our other step here. We actually will, in between here, have to wash our cells. And the reason we have to wash our cells is, remember, in this patient plasma here, okay, if we make our big picture of our drop of plasma over here, there are other antibodies in here. Remember, our chickenpox antibody is in here. Our COVID antibody is in here. Our mono antibody is in here. Our flu vaccine antibody is in here. So there's all sorts of antibodies in here. And so we only want to detect antibodies that are on the red blood cells. So we add this, we incubate it, okay, and then we wash the cells. Okay? So we're going to wash the cells, and in essence, when we are washing those cells, we are removing all of those other antibodies in here. Okay? Because if they stayed in there, they would neutralize and give us a false negative if we didn't wash. So we're going to wash them. And what we're going to have left over here, I'll draw my antibodies first. Okay, that's what we have left after washing. All of these guys here have washed and gone down the drain. Okay, there you are. Down the sewer system. Essentially, exactly, that's where they've went. Okay. The next thing we're going to do is we're going to add now our reagent, our IgG, our AHD. And what that's going to do is, if these are in here, we're going to cause this lattice formation, and we'll see agglutination, and we'll be able to detect those antibodies. Okay. We use this technique, this IAT, or indirect antiglobulin test. We're going to use it for our antibody screen. We'll use it to identify those specific antibodies. If we have an antibody, we have to use it for cross-matching, and we may need to use it for antigen typing. And what antigen typing is it identifies these specific antigens on patient or donor cells. Okay? This cross-match, this is what we do when we take the unit of blood we're going to give to the patient. Okay, this unit of blood, and we mix it with the patient's plasma to make sure there's no reactions in the tube. Okay, and we'll talk more about these throughout the course as well. Okay, let's talk about an antibody screening test right now and define what it is and what we use it for. Okay, so an antibody screening test, you are going to use specific screening cells with this one. Okay, and they are usually screening cell 1, screening cell 2, and some sites are going to use 3. So there are any variation of 2 to 3 cells. And what we utilize them for is to determine if a patient has any unexpected antibodies in their plasma. So there are antibodies that we expect in a patient's plasma. For example, if the patient is type O pos, okay, we expect them to have anti-A and anti-B. So those are expected antibodies. You know, if the patient is A, we expect them to have anti-B. And if they're B, we expect them to have anti-A. And if they're AB, well, that's actually when we don't expect them to have any. Okay? So these right here, these are your expected antibodies. What we're looking for in this test is any unexpected ones. And how do you get an unexpected antibody? And this is where you'll get an unexpected antibody if you have been previously transfused or pregnant. Okay? And in those instances, whoops, sorry. Did I spell that? I think I might have spelled that right. That's okay. P-R-E-G, A-N-T. I had it right. So if you, this is how you're exposed to foreign red blood cell antigens. Okay? And you're exposed to these foreign red blood cell antigens by being transfused or if you're pregnant. And when you're exposed to these other red blood cell antigens, if they are different than you, okay, they have, let's put Kel on this one. We're going to go with big K or Kel. So if this is happy old me, we'll just say it's me at the moment, okay? And I don't have Kel, okay? I don't have Kel on my red blood cells, Okay, just like I'm O, we know I'm O positive. Okay, so I don't have A and B antigens on my red blood cells. And we're going to say I don't have Kel on mine either. So my red blood cells are just these happy little blank slates, so to speak. They're not. We'll determine what other things are on mine later. But so these are my red blood cells. Okay, and now over here, you have this red blood cell with a Kel on it, and I need blood. Okay, so you're going to give that to me. Okay. Well, now, once this red blood cell with this Kel on it is circulating with my, the reason I'm being transfused is I don't have a lot of my happy little blank slates. Okay. But now I have this foreign red blood cell with this foreign antigen on it. Okay. So what's my body going to do? It's going to recognize that as something that's not me. And so I am going to then start producing an antibody against Kel. Okay. Now this time right here, this is my first exposure. When I first get this, it's not going to be a big deal. It's not really going to cause any problems. Okay, and that's what we talk about, first exposure. But the second time, if I'm to get those red blood cells again after I've made the antibody, that's when we're going to have issues. And so this is where this antibody screening test comes in. This is why it's important. So I'm happy for a little while. Okay, you can see I'm happy for a little bit. And then as time progresses, I become anemic again. So here I am. I'm not feeling so great. All right, I have just a couple pale looking red blood cells. So I need another transfusion. So they're going to do an antibody screen. So now when they draw my blood, okay, they're going to draw my blood and they're going to test my plasma. Here's my plasma. They're going to see and look to see if I have any unexpected. Okay, remember, they're expecting, because I'm O, they're expecting the anti-A and B. But now they're going to test my plasma in at least 2 of these with screening cells 1 and 2. Okay, just to see if I have anything. Okay, initially I didn't have any unexpected antibodies, but now we're looking to see this. And notice I am drawing these. This is an IgG. Okay? And remember, this is an IgG antibody. We can't detect this, just an initial spin, like we can when we're testing for anti-A and anti-B. Okay? So we have to do something additional to this. So what we're doing is we do all of these phases. Okay? This is our first phase, which is immediate spin, like what you would do for your back type when you're looking to see if the patient has anti-A or anti-B. So that's our immediate spin phase. So we take these tubes, we add this plasma, okay, and we add screening cells 1 to this one. This is our reagent. Remember, we have to have a known thing. And then these are screening cells 2. I'll make them just look a little bigger. Okay, those are screening cells 2. And then we add my plasma. How many drops? Remember, it's always 2 drops plasma or serum to 1 drop cells. Okay, so we add that. We do immediate spin. We read it. Okay, we record our results. Next, what we're going to do, because this is IgG, and what temperature does IgG like? Not room temperature, which is what immediate spin is. Okay, so now we're going to do an incubation phase. Okay, this is now going to be our second one. So we're going to incubate it at 37 degrees Celsius. Okay, we'll use 10 to 30 minutes if we're adding enhancement, and pretty much everyone uses enhancement media because we don't have time to wait 60 minutes if we're not using enhancement media. Okay, so we even use it in class. So we're cutting our time down by at least half, okay, sometimes by 1/6
And that's really significant, especially if you have a patient in the emergency room or surgery or something like that. So after this incubation phase, we're going to take our tubes and we're going to centrifuge them, and we're going to read again for any reactions. But remember, this is IgG. Okay? IgG, if it is in there, it is not able to form that linkage and make that lattice formation so that we can visualize that agglutination in the cells. So after the incubation, after we've read for reactions, okay, we are going to, this is where that washing step comes in. We're going to wash 3 times, and then we add the anti-human globulin. Okay. This anti-human globulin is a reagent. Remember, it's green, and it is that antibody against human antibodies. I know that always throws us off. Okay? So it's an antibody against human antibodies. So what it does when it's in there, let's draw a bigger picture. We're going to go over this as many times as we need to. Okay? So go right here. I'll just draw a slightly larger tube. Here's our red blood cell. Okay? We're going to put this anti-kel on it. Remember, he's kind of wimpy, he can't form that bridge. So now we're going to add the anti-human globulin, drop of reagent, and because this is against this, okay, it's going to bind to these, and it's going to cause that bridging now, that lattice formation, so we can see agglutination. And so this is, remember, this is also indirect, indirect antiglobulin. Okay. And now we'll see this agglutination. But again, these are still a little wimpy, and they are not IgM. So IgM is amazing, okay? And we get 4+ reactions when we have good IgM. Okay? When we add this anti-human globulin, we're usually not typically going to see 4+ reactions. 2 to 3+ reactions are going to be what you normally see in this, if it's positive. And remember, again, our goal is no antibodies, okay? We don't want to find antibodies, but we can, okay? And it happens, not a lot, but you will find some. Okay? So that's our antibody screening test. Okay, now we're going to talk about our check cells. And I want you to remember, this is blood bank, like you forgot, right? Okay. We are in blood bank. And what do we tend to do when we work in blood bank? Okay. We double check everything we do. Okay. We don't want to make any mistakes. Okay. So we have a lot of checks and balances just really built in. And notice this says check. Okay. These cells actually have several different names. And so check cells, you may hear Coombs control cells. This is really a very old term, and I've even heard individuals, when we talk about the anti-human globulin, or AHG, they have called this Coombs reagent. Okay. I do not tend to use this term. It's definitely one of the older terms. Okay, but that's something you might hear. So that's where these
Coombs control cells come. Okay. And what exactly are these cells? So let's talk about what they are. And this is a reagent vial that has red blood cells in it that have human globulin or complement. Okay, so IgG, human globulin, or complement all over them. So they have antibodies already all over the cells. Okay. And why? We can put little C's on this one for complement. Okay. And why? Well, these are controls. So what we're going to use them for is to verify. Because remember, we have to verify, we want to verify everything that we've done, all of our testing has been correct. Okay. So these are also, we can call them, they're D positive cells coated with human anti-D. Same thing. Okay. And what we're really looking for, if they don't react, we want to see a reaction. It means we've not done something correctly in our testing protocol. And typically it's improper washing. And this is, let's draw some pictures here so we kind of understand. So this would be our test tube that we were doing our testing in. Okay. And here's our reagent cells. We'll just call these screening cells 1. Okay. So these are our screening cells in here. But we use this anytime we use AHG in a test. Okay, not just screening, but you'll see. Okay, so here's our testing protocol, and we added our patient plasma. Okay, we added it in here. There's our patient plasma, full of all sorts of antibodies that we would expect. And what we're trying to determine is if there's any in there that we didn't expect, unexpected antibodies. So we've added it, incubated it, washed it, I hope, okay, tested it, and in the end we have a negative result. Okay, and so let's add this in here. And it didn't bind to anything. Whoops, we just want to make this human globulin. Okay. It didn't bind to anything because I didn't have any unexpected antibodies. There's nothing on these red blood cells here. Okay. See, there's nothing on the outside of them, so I didn't have any unexpected antibodies. Okay. So this anti-human globulin had nothing to bind to. So I have a negative result at the end of my testing. So I'm going to add these cells here, bringing them down here, and I'm going to add them to this test tube. And when I add them to this test tube and I centrifuge it, and remember when I centrifuge it, I pull all of these cells close together down at the bottom, okay, so that they're close together so they can potentially react. And if I did everything properly, okay, that those check cells will actually react with the anti-human globulin. Okay, so let's put it back in there. We'll put our anti-human globulin in here. There it is. Oh, I picked a different green. Sorry about that. Okay. And let's go back and let's get these cells. So if we have these cells here and they had that anti-D or immunoglobulin all over it, okay, that anti-human globulin is now attaching to it and causing agglutination that we can see. Okay, so these are used to make sure that anti-human globulin is in there and still active and it works. So we should get, you know, roughly a 2+ reaction when we add this. Now, if we don't get a reaction, it means we didn't wash appropriately. And why do we care? Okay, what kind of problem does that cause? And so if we didn't wash appropriately, this is what happens. Let's go back here. So here's a tube we're going to draw. And here's the bad scenario, okay? This is not what we want to happen. So we're doing our testing protocol. Here's our screening cells in here. Okay. And here's our patient plasma that we dropped in here. All right. And remember, we have all sorts of antibodies in our plasma. So there's our chickenpox antibody. Here's our COVID. Here's our influenza. We have a lot of them floating around in our plasma. Okay. Now, we incubated this and we are supposed to wash. When we go through the washing step, okay, that would essentially remove all of these and they would go away. They wouldn't be in here at all. Okay. That's what happens in a proper washing protocol. If, however, we do not wash appropriately, let's see, I want to go back. So we don't wash appropriately. When we don't wash appropriately, these antibodies stay in here. Okay? So this is the improper scenario. And when that occurs, and we now add our anti-human globulin, here's our anti-human globulin, we're adding to it. Remember, these guys here are human antibodies. So our anti-human globulin is going to bind to any human antibody. And so what happens is if we don't wash these away, this antibody is going to bind to these extra ones that we don't care about. Well, if they bind to the extra ones we don't care about, there's none left to bind to the ones that we might actually care about. Okay? So it's essentially used all of our anti-human globulin up, and we will get a false negative. And that would be bad. Okay. Now, how do we verify that we've done everything properly? This is where your check cells come in. Okay. Whoops. This is where our check cells come in. So we would add our check cells right here. Let's see if I can do this a little easier here. Okay. We're going to add them. There's our check cells. Okay. They're now in there. Okay. Because that human globulin, this human globulin, is bound to this human globulin, it now can't even bind to the check cells. Whoops, I didn't want to use that. Okay. So it can't bind here like we'd like it to. So if it's all neutralized, it can't bind, and we're going to get a negative result. So instead of getting this 2+ like we did here, we're going to get a 0 because this human globulin here has been neutralized. It's been neutralized by these antibodies that we didn't wash away. And so now it's not available to bind to our check cells, and we will not see that agglutination. And so we know at that point in time the test is invalid, and we have to start over. So if your check cells are ever negative for your check cells, that's an invalid test, and you have to redo it. Okay. So here's just a box from the book describing
consolidating what could be some reasons why our check cells didn't work. So what are our check cells again? They are red blood cells coated with human Ig or complement. Okay. They are added to all negative reactions in a tube-based AHG testing. Okay, so all testing that you use AHG with, you have to add check cells. Okay, and what does it do? It validates the test, okay, because if there was any free AHG, okay, it's going to bind with those cells and it'll give us at least a 2+ reaction. Okay. It causes that agglutination. Now, the problem is if when we add our check cells, they're negative, we've got to repeat the test because that is an invalid test. Okay. Just think about your flu test when you put the drops onto there, the well, and they migrate across here, and we have a control line and we have a test line. Okay, you always have to have, whoops, sorry, I did that backwards. You always have to have a line at your control line. Okay, and that's exactly how this is as well. You always have to have a positive when you add your check cells. Okay, if it is negative though and it is invalid, why? We want to know why. Well, occasionally you might get distracted because you're in blood bank and it can get kind of crazy. You forgot to add AHG. Well, it's green. Okay, it didn't used to be green, but it is now to help us prevent this. So it's green to help us visualize that we did add it to the test. Okay, your reagent could be bad. So maybe it is green, maybe you did add it and it's still not working, and maybe it's bad. So maybe you need to repeat the test with different AHG. But honestly, the most common issue is inadequate washing. Okay. And that's kind of your classic issue with it, classic test question, and so forth. Okay. Enhancement reagents. So we have talked a little bit about enhancement, and what it accomplishes is it enhances the reaction, okay? Decreases the incubation time, thank goodness, right? The incubation time. Okay. But what it does is it increases the rate, okay, and sensitivity of the antibody attachment to the red blood cells. What are some of our enhancement medias? We have albumin. Okay. I do want you guys to know these. If you're asked, maybe you'll have a question that says pick all of the enhancement medias, or which ones are enhancement medias. You need to know this. LIS, which is low ionic strength solution, and PEG, which is polyethylene glycol. So these are the three most common enhancement medias or reagents used in the blood bank testing. Factors affecting our anti-human globulin phase are the same ones that we've been talking about that affects all of our testing. Temperature. And one thing we'll talk about with temperature, we do the immediate spin phase, and when we're in immediate spin, that's where we're detecting IgM antibodies. Okay. We do our 37 degrees, because this is where our IgG antibodies react and they're attaching, and then we do our AHG phase. Okay. And this is again where we're detecting the IgG antibodies. We are not seeing reactions here and here with antibodies that are IgM. Okay? And here we're not seeing reactions with antibodies that are IgG. Okay? These phases are very specific
to these antibodies. Okay. Ionic strength. Okay, this is where we have all of those little charges in there, and they're either working together to push things away, or we're altering them a little bit to try to bring things together, closer together. Okay. Plasma to cell, whoops, that's my highlighter back. Here's our plasma to cell ratio. And remember what this is. This is 2 to 1. 2 plasma, 1 cell. Okay. We can draw it like this. I know my drops aren't very good. To 1 cell. 2, whoops, I went in red. There we go. 2 to 1. Okay. Because if we have an excess of either, what happens is if we have 2 little cells, okay, and then our antibodies just, you know, cover it all up, and then we never get any agglutination. Okay. If we have too many cells, then we don't have enough antibodies. Okay. Again, it's both ways. Okay. Incubation time, depending on our enhancement media, if we don't give those antibodies, antigens, time to actually stabilize with that attachment, we won't find it. Okay. And pH. All right. Needs to be at the proper pH so that those cells want to move together. So in our next slides, we're going to be moving on to the DAT. So I'm going to end this one for now, okay? And we'll have a separate one for our DAT. But for right now, that is our indirect anti-globulin testing. So our IAT. Okay. Thanks, guys. Signing off.